first thing today, Cecilia wanted me to show you this, so I'm going to let her go ahead and plug the micro club.
Hi guys, it's Cecilia. So I don't know if a few of you may or may not be aware, we do have a micro club on campus. We're the student chapter of the American Society of Microbiology. And like everything else, we do meet virtually, don't meet in person. But basically the club consists of us discussing a new microbiology topic every other week. We have discussions. We're hoping to get some guest speakers. Overall, it's just a cool place for us to meet and build community. You know, it's really hard to do that during these times, and I feel like it's a good opportunity for us to feel more connected as micro majors, or even if you're not a micro major, we're very inclusive. So yeah, our first meeting is this Friday at 3 p.m. If you want more information, we're going to be doing like a little intro session where we can all meet and talk about what the club is about. So I hope you can make it. Do you guys have any questions? So yeah, if you want to come, feel free to take a picture. You can also find us on Instagram at our little handles right there, but hope you guys can make it.
And I'll post the flyer on the Beachboard site for the class too.
Thank you.
So, all right, sounds good. Okay, so last time we didn't quite finish the second lecture, so we're going to go ahead and finish that one off first, and then we'll move on to today's ecophysiology. So one of the things that I hope I impressed upon you last time was that there was this large amount of diversity, and most of it is untapped or unknown in the microbial realm, whether that's bacteria, archaea, or even the microbial eukaryote fraction. And so one obvious question is, well, how the heck did we end up with this much diversity? And what we need to think about is that, first of all, microbes, you know, we expect that the first life on Earth were not zebras and redwood trees, right? So we know that they were likely microbial. How they exactly relate to modern microbes, we don't know, as we talked about last time, but we know microbes have essentially been evolving for probably at least 3.8 billion years. So that's a long time. That's a lot of opportunity for them to evolve into the very vast diversity that we see today. The other thing to note or think about is their generation time and what that means for their evolutionary rate, right? So mutations, right, are our major source of sort of raw material for evolution to act upon. And if you remember from your general bio or your genetics class, right, the majority of mutations are going to be neutral or deleterious or negative, but every once in a while we get these positive mutations which may become selected for, fixed, often in communities, particularly microbial communities. But these mutations arise at a fairly steady rate. And what is the major cause of mutations in most organisms? When does that happen in an organism's life cycle? Does anybody remember?
The reproduction.
That's right. So during reproduction, and specifically during what?
During the recombination of
meiosis.
So, well, not meiosis, right? Do we have meiosis with bacteria? No.
No, binary fission, my bad.
Yeah, so, and it's not so much, what I'm really looking for here is DNA replication, right? So recombination can occur in a number of ways after the fact, but the actual origin of a specific mutation, right, like a base change from A to C or whatever, is typically a replication error. So this occurs every time a cell is going to divide. And so the short generation times allow that to happen more frequently. Right? Elephant mutational rates are much lower than bacillus mutation rates, right? And so their evolution can be more rapid, right? And so this just shows you So most of them are going to either be neutral, right, which means that natural selection doesn't impact them one way or another, or negative. And if it's a negative, it's likely to be selected out, right? That organism's likely to die or reproduce more slowly, and so it will be lost over time. But if it's beneficial, maybe 1 in a million mutations is beneficial. But what often happens, and this has been posited by actually a former instructor of mine, former professor of mine, Fred Cohen. So he's talked about selective sweeps. He's not the only one, but he really argued in this 2008 paper for the importance of positive selection for, like, essentially, it's like a replacement, or what we'll talk about later in the class is a sweeps through, and all of the relatives that don't have that mutation will be outcompeted and die out. This is kind of analogous to what we're seeing with some of these virus variants that are emerging in COVID, right? So we're worried about these rapid spreaders because they're likely to replace the more slowly spreading virus that's out there. And the same kind of thing can happen in bacteria and archaea. Okay, so prokaryotes have evolved into many different niches, and we're going to talk about those niches in today's lecture. Okay, and we're going to talk a lot over the course of the semester. One thing you need to remember, and we'll touch on this in the second half of the class also, is no two organisms are so-called species, right? Which is sort of a tough definition. They're exactly the same ecological niche. If they did, they would be competing with each other completely, and one would likely outcompete the other. So what we've had happen over many billions of years is this huge metabolic and functional diversity. And so what that allows is they differentiate each other even just a little bit, or maybe they differentiate each other in when they're active, where they live. All these different environmental factors can play in and allow this diversity to exist, right? And then we can mix this up even further if we introduce lateral transfer through processes like conjugation, where plasmids are transferred, or transformation, where naked DNA is transferred, or transduction, where viruses mediate the movement of genes from one organism to another. And this can even occur across domains. We have viral genes in us, we have bacterial genes in us, we have archaeal genes in us, and vice versa. Because over the billions of years of evolution, genes get moved around a lot, and this contributes to the diversity. And this just highlights this. This is a paper from a few years ago, and what they did is they sequenced over 100 archaeal genomes, and they compared those to over 1,800 bacterial genomes, and they found evidence that there's 5 times more lateral gene transfer of genes from bacteria to archaea. So it goes both ways, but it seems like the majority of the lateral transfer is going from bacteria into the archaea. And oftentimes these are metabolic genes that are moved, and they can lead to cladogenesis, or what's referred to as a new, like, speciation event, or a new group even, maybe more than one species, and diversification in the archaeal TOL, which just stands for tree of life. So it may be that the diversity of archaea we see now is somewhat related to the fact that there's a lot of diverse bacterial genes being moved in through these horizontal gene transfer events, even across domains, which is pretty phenomenal if you think about it, right? So we would never think of a plant gene being inserted into us and working, right? But like geneticists, when they do genetic manipulations, they usually look for close relatives, right? But we are much closer related to plants than bacteria are to archaea, and yet they can exchange genes, and most times it might not be beneficial, it might even be a negative, but every once in a while you end up with this movement that is selectively positive, which then can lead to these diversification events. Does that make sense? Any questions about that? I'm always astounded though that that's possible. All right, so let's have our first poll question. So I told you we don't know what happened at the origins of life, but based on what I've talked about and what you've learned in other classes, which type of organism do you think came first? There's no right or wrong answer for this. We'll just talk about it. Whoops. So I'm going to launch the poll. So which of these four, don't put E, please. Chemoautotrophs, photoautotrophs, photoheterotrophs, or chemoheterotrophs. Hopefully you guys remember those definitions that we kind of alluded to last time. Which do you think came first? Take a guess and then we'll talk about it. All right, there's a few more of you. Just go ahead and log an answer if you haven't already. Okay. Two more. You don't lose points for guessing wrong. I guess we have, okay, that's good, we're good. All right, so it looks like most people are evenly split between A and B. And so everybody, the vast majority of you are voting for an autotroph. Somebody tell us why you picked autotroph. Why would that be? And then maybe explain why you picked either chemo or photo. Anybody want to be brave?
I have an answer. Yeah, so
Okay,
for,
go ahead, Jose.
well, to narrow it down to at least the autotroph, I kind of thought it's not like the chicken or the egg came first. So it's like, in order to be a heterotroph, we would have to be kind of like relying on other things to be in place in the environment. And if it was in autotroph, then it doesn't necessarily need something else to get that process going for it.
Yeah, I think that makes a lot of sense, Jose. I mean, there is organic, there are organic molecules out there to be had in the environment. There's actually organic carbon. We talked about you could make it in the prebiotic soup potentially. But I think a lot of people think the autotrophy probably came first. What do you think in terms of chemo or photo? I don't know which you put, but you want to share?
Me?
Sure.
Oh, yeah.
What did you guess?
I put chemoautotrophic.
Okay, and why?
Just based off like the last lecture, we talked about how there's a lot of like, well, I don't know if it's the correct term, but like electron donors. So we have hydrogen available and everything. And I would say like, in order for it to be photoautotrophic, you would need a lot more, actually, no, never mind. Sorry, I was gonna say something about oxygen, but lost my train of thought.
Yeah, they produce oxygen. They don't necessarily need oxygen, but yeah, and Jenny shares that she thinks chemo because of the early conditions on Earth. And remember, UV was really high on the early Earth, and so that might argue against photosynthesis being the first metabolism. Chemosynthesis, it was hot, like hydrothermal vent environments where chemosynthesis or chemoautotrophy is important. Might have been, we talked about hydrothermal vents as being a possible cradle of life. So yeah, we don't know though, right? So I want to emphasize that we really don't. We don't know. But here is a possible, right? And this is kind of conservative. We do have evidence of chemoautotrophy of almost 3 billion years. And then you might imagine that there's photoautotrophy. And as we're going to learn later in the course, earliest photosynthesis did not produce oxygen. And there are plenty of bacteria today that are anoxygenic phototrophs. And then later, maybe about 2.5 billion years ago, oxygenic phototrophs came in. And then you could plausibly see maybe chemoorganotrophy coming in. Okay, so this is one possible scenario, but we don't know for sure. But I think it makes sense. And Jose, I think you kind of hit on a lot of the reasons why we think autotrophy might precede heterotrophy. And based on the sort of evidence from the fossil record, we think oxygen production didn't start at the beginning. Okay, cool. All right, so that is the end of the last lecture. Any questions about that? Okay, so go ahead and start up our next lecture on ecophysiology. So this really dovetails in very nicely with the discussion we just had about metabolism. So we're going to just talk and start to talk about how microbes grow and things like that. And you guys, at least well, even non-micro majors should be aware of kind of like what a bacterial growth curve might look like, right? And in a culture flask, many of you have grown bacteria on a plate or in a culture flask. What do we typically see in terms of the growth pattern of something in a pure culture? Does anybody want to share that?
It's exponential.
Okay, is it exponential right away?
No.
Okay, so what are those phases you guys learned about? Somebody want to help, Carlos?
Log phase, lag phase, stationary.
Yeah, so, right, you guys have all seen this, right? We even show this, I think, in general bio, right? So there's that lag, and then there's this really steep exponential growth usually. And then stationary phase, what's going on in stationary phase? Does anybody remember? Why do they stop growing exponentially?
Because they run out of resources to continue to grow.
Right, so they run out of something. It may even be one thing, right?
Mhm.
But if you're missing that single reagent or nutrient or whatever, you're done. And so it just kind of flattens out. And then in some cases you go into death phase. I will say in my own experience, right, because you're going to measure this by like reading in a spectrophotometer, reading the optical density over time. And in many cases, this is more of a gradual decay. And real death phase may not even show up in a lot of organisms. Okay, now my next question though is, how relevant do you think this is in the environment?
It's not really an ideal representation.
Okay, in what ways? Like what do you think the distinction is or difference would be in the environment?
They don't have ideal or optimal conditions, so maybe they don't have all the nutrients necessary.
Absolutely. So out in the real world, it's cold and dark, literally, right? And food may be limiting. The conditions will not be anywhere near what you would do if you're growing like E. coli at 37 degrees, which is like human body temperature, right? In an incubator, and you're feeding them like more food than many organisms in the environment will ever see in their entire lifetimes, right? So the thing to really think about is that this is going to change our curve. So the lag is typically much longer. The log phase, if it even happens at all, will be much shorter and probably less steep. Because remember, the steepness of that curve is related to how high the cell density can get. And so your what we call the carrying capacity, and we're going to touch on this idea later in the semester, may be much lower in the real world, okay? And then many organisms will be in stationary phase, or what you might call death phase, or in some sort of dormancy situation the majority of the time, right? So they're just kind of like sitting there waiting for food or water or light or something like that in their environment to change so that they can then start growing. So it's a very different environment out there. Okay, any questions about that? All right. So there's this term, we're going to have a couple terms today, otecology and synecology. So the first is otecology, which is the study of microorganisms in their environment. And so today we're really going to be sort of exploring this and thinking about it. And if you're a macro eukaryote like us, right, this tends to affect us at the individual level, right? Do I have food to fuel my body? That's, you know, it may be a collective food kind of thing, but in general, what we're talking about is, you know, this human being ate, and so they survived that day, right? If they're starving, they may die over a course of days, weeks, months, whatever, but the individual is really where we think about this. And this is true for all sorts of environmental impacts, right? Do we have oxygen to breathe? Do we, you know, have heat stroke that kills us or whatever, you know, these kinds of things. For microbes, though, we're often going to be viewing these and many concepts that we'll talk about this semester in terms of population effects. Because remember, if you think about like a microbial colony on a plate, how many cells do you think are in a colony that might form on a petri dish? Does anybody have a sense? Is it tens, hundreds, thousands, millions? What do you guys think?
Thousands.
It's for sure at least thousands. In many cases, it may be millions of cells in that colony that you see on your plate, depending on how large it gets, of course, right? So if you think about it, yes, the cells on the outside of the colony might be experiencing a different environment, those in the middle. But when we assay them as ecologists, we're probably going to treat that colony as a single unit. And it makes sense for microbes because that colony arose from how many cells?
One.
Just one.
In most cases, we would assume it arose from one. Now, you might have overgrowing colonies, but we're going to ignore that for this discussion, right? So all of them, if they arose via what kind of reproduction? Like if it's bacteria?
Asexual.
So if it arose by asexual, like binary fission, right? They're all identical clones. And so we can really think about the interaction of those organisms with their environment as like a whole, but it's really the population responding to it. Does that make sense? So that's often what we're going to be looking at is population level effects. Whereas with so-called higher organisms, we often look at individual effects. Oops. Okay, so the next poll question. Oops, I got rid of my poll, I think. Dang it. Mouse does not want to. Here we go. All right, I will relaunch the poll. So which of the following would limit growth or survival of microbes in nature? Reduced nitrogen, the presence of other microbes, an increase of temperature, both A and C, or E, all of the above. Which do you think of those could impact their survival out in nature? Okay. Maybe one more. Excellent. Okay, so when I do the sharing, can you guys see the poll results?
Yes.
Yes.
Okay, good. I thought you could, but I couldn't remember. It's been a while since I've been doing these polls since. All right, so you can see most of you put E, which I would agree with. So really all the rest of you that put these other answers did technically have a correct answer. But let's go through them individually so we can understand why it really is all of them. What do you think, somebody who thought A was a correct answer, why would that be something that might limit? Why does that nitrogen matter? Do you need nitrogen to live? See, someone's responding in the chat. Jenny is responding in the chat saying metabolism. Okay, so some organisms actually use nitrogen for their metabolism, like they might oxidize or reduce nitrogen and make a living that way. Okay, but not all organisms do that, right? Caden says, doesn't nitrogen help produce nutrients by breaking down organic products? So organic products actually may contain nitrogen in them, okay? And so if organic products break down, then they may release nitrogen into the environment, if that's kind of what you were getting at, Caden. Alessandra brings up nitrogen fixers, right? So nitrogen fixing bacteria are an example of organisms that are metabolically nitrogen users, right? And so there are those specialized organisms like Rhizobium. Yes, Hamza, that's an example of a nitrogen fixer, okay? But we all, they all, and we all need nitrogen for some basic cellular functions. What do we need nitrogen for? Where do you have nitrogen in your cells?
DNA?
DNA. That's pretty fundamental, isn't it? Where else do you have nitrogen in your cells? Also very fundamental. So if I say that remember your chemistry and that a nitrogen, an NH3 group is called an amino group, would that help you? So what macromolecules have nitrogen then?
Protein.
Proteins, right? So two of the fundamental building blocks of life require nitrogen. Yeah, so thank you, Jennifer. I saw that you also put that in there too. Yes, so amino acids and Amy as well. Good. Okay, so yes, so nitrogen plays a big role, okay? The presence of other microbes, how might that limit growth or survival?
Competition.
Exactly. Yeah. So why does competition limit growth? How does that work?
So a population might take over all the resources and then leading to deprivation for other population and their death.
Exactly. And as we'll learn about later in the course, competition also occurs within a population, right? So if you think about that growth curve we just saw earlier, when you go from log phase into stationary phase, essentially that is the result of intra-specific competition. Something gets used up. The brothers and sisters cells in that flask are competing with each other and they run out of something. Very good. Okay, increase in temperature. I think this one was pretty obvious. How would an increase in temperature potentially limit growth or survival? Somebody maybe who hasn't said anything yet? You can share in the, Robin in the chat says optimal temps increase growth. So you're absolutely right, Robin. You can increase growth if you increase temperature to a certain point. But here we're really talking about limiting growth or reducing growth. So when might that occur?
It kills them.
Say again?
It can kill them by denaturing the proteins in their system.
Yeah, Daniel. So if you exceed their optimal growth temperatures, right, then oftentimes what's happening is proteins are denaturing. If it gets really hot, DNA can unwind, right? Cells can lyse, these kinds of things. So it's a fine line because increasing temperature can actually speed metabolism, speed growth. But then if you exceed that, then it can definitely be in this category of limiting. So all three of these were correct answers. And so E was the best answer. Does everybody see that now? Awesome. Okay. So let's talk about some of these conditions. I've introduced a few of these in this, but there's certainly more, okay? And so I'd like us to explore it.